everyone, today the video will be focusing on fructose 2,6-bisphosphate regulation. So our liver cells need to regulate and maintain our blood glucose levels. These cells have to regulate glycolysis and gluconeogenesis in a reciprocal manner to ensure that the proper glucose levels are maintained. Within this reciprocal regulatory pathway, a key element is a bifunctional allosteric enzyme that contains two different regulatory domains. And so one domain is called phosphofructokinase 2, or PFK2, while the other is called fructobisphosphotase 2, or FBPase 2. And so in the unphosphorylated state, the PFK2 domain is active while the FBPase2 domain is inhibited. And so PFK2 is going to work by converting fructose 6-phosphate into fructose 2,6-bisphosphate. Now, in the phosphorylated state, PFK2 is inactive while the FBPase2 domain is active. So this is going to be phosphorylated. So in this state, PFK2 cannot stimulate fructose 2,6-bisphosphate production, and FBPase2 is going to cause the dephosphorylation of fructose 2,6-bisphosphate into fructose 6-phosphate. So it's important to remember that FBPase2 and PFK2 are the same enzyme, but they respond differently to phosphorylation by protein kinase A. So let's look a little bit more into the fasting state. In the fasting state, we have high glucagon. And so that is going to increase the amount of cyclic AMP through a G-coupled protein receptor. And this is now going to increase the activity of protein kinase A. And so because we have high levels of protein kinase A, this is going to now phosphorylate FBPase2. So this has become active and our PFK2 domain is inhibited. And so now we're going to convert fructose 2,6-bisphosphate into fructose 6-phosphate, and gluconeogenesis will proceed and replenish blood glucose levels. In the fed state, we have high levels of insulin, which is going to cause a MAP kinase cascade to occur, which is eventually going to phosphorylate something called phosphoprotein phosphatase 1, otherwise known as PP1. And so PP1 is then going to dephosphorylate FBPase2, and it's going to inhibit FBPase2 while simultaneously activating PFK2. And so now the body is going to convert fructose 6-phosphate into fructose 2,6-bisphosphate. And so fructose 2,6-bisphosphate is actually going to help activate PFK1. And so PFK1 being active means that glycolysis is going to occur because it's going to take fructose 6-phosphate and convert it into fructose 1,6-bisphosphate. It's important to note that fructose 2,6-bisphosphate is created to regulate glycolysis, and that PFK1 is a committed step in glycolysis. So it's important to regulate this enzyme more than other enzymes within the pathway. Other regulators of PFK1 are ATP and citrate, which are going to inhibit this enzyme, while AMP and F26BP are going to stimulate PFK1. And so ATP is going to inhibit PFK1 because you don't want to promote glycolysis to make more ATP if you already have a lot. Hey, AMP will actually inhibit FBPase1, but it will stimulate PFK1 for that same reason. Most importantly, F26BP will inhibit FBPase1 but will stimulate PFK1. So F26BP is going to promote glycolysis, but it's going to inhibit gluconeogenesis. I hope this video was helpful in learning more about fructose 2,6-bisphosphate and its regulation within glycolysis and gluconeogenesis. Thank you.